Can we confirm that higher amounts of nitrogen, overfertilization rates of nitrogen, can lead to higher aphid problems? Yes. Should we try to reduce the nitrogen status of our crops in order to reduce the aphid issues? Probably not, because it's unlikely that you can do that without impairing the crop's growth. Like, who cares if you have aphids if your crop sucks?
This is Serious Grower Podcast. I'm Anna. I'm an entomologist. I'm a scientist, and I'm using this podcast to talk to all kinds of growers and eaters who are serious about their farms, their gardens, their local food systems, and I'm asking, how can science help? Today's co-host is Caleb Goossen. Caleb is an educator and a scientist, and in our neck of the woods, he's kind of a celebrity, so you might recognize his name if you follow him on Instagram at Organic Crop Specialist or subscribe to his newsletter from Maine Organic Farmers and Gardeners Association. We call it MOFGA. Now, I have considered Caleb a friend and colleague for so long now that I kind of forget we've only met in real life a few times. I think a lot of us can relate to these post-pandemic professional working relationships that only occur over Zoom. Just the other day, I was feeling kind of excited because I got to meet Caleb's family. And then I had to remind myself, now, Anna, you just waved at them over a Zoom call. But, I guess that still counts. What a time to be alive. All that to say, today's episode may or may not be for you, depending on how much you heard about plant sap and its connections to pest severity. A lot of you might be thinking, what gives? But I know a lot of you out there, a lot of you serious growers, might be thinking, yes, finally, I have been dying to hear what Caleb thinks about plant sap, myself included. So here we go. First of all, I want you to clarify, what is the claim? Because it seems like the people who are putting forth this claim, they have good intentions, right? Like, it's good faith. They're relying on science. The claim is you can manipulate your fertilization strategy in such a way that you don't have to worry about pests.
Or you can reduce the impact of pests, or multiple versions of something along those lines.
I've only peripherally been paying attention to this because this is definitely... it's getting at a phenomenon that we know to be true about certain groups of insects, but I don't know if I really understand how part of the zeitgeist for growers this is. Is this something people are talking about? Is this something you're getting a lot of questions about?
I wouldn't say I get a lot of questions about it, but I think that's also because the people who are thinking about these things are taking them for granted. There are a lot of folks who bring up conversations about growing with me with some assumptions about things that they just assume that I'm thinking the same things or through the same things or have read the same books or heard the same people speak that they have. There's sort of two layers, I guess. There's the one layer, which is every fact sheet you've ever read about aphids in particular mentions that high nitrogen fertilization can lead to increased aphid issues. Sometimes they attribute that to things like rapid lush growth that's a little softer and easier for them to get at. There's also been different approaches to fertility management, particularly for commercial farmers, but it also crosses over to any grower. There's long-standing history of folks who are really into what they talk about balancing. They're really into micronutrients. Issues that they see, disease, pest, other production issues, all get traced back to, well, then something's out of whack. There's some sort of misbalance, and they're looking for sort of a divine balance that should be there. And if you could reach that balance, then everything is easy. That's oversimplifying, of course.
And that's why it's such an interesting topic of discussion, because there's so much good stuff in here. But then there are some stretches and things like that where you go, oh, whoa, whoa, whoa. I don't want to say dangerous. I think the thing that bothers me a little bit about it is that there's a little bit of if only you knew how to do this right, you wouldn't have to worry about pests, right? If somebody is either just starting out or they're struggling with something and they say, oh, if only I got it right, I wouldn't have to worry about pests. That's the not cool part about it.
Yeah, so I'm going to go in two directions here. One is I do try to stop and think once in a while of does fill in the blank practice matter versus what I would normally suggest? Because sometimes, even if the rationale behind it doesn't make sense to me, And even if I don't expect it to be particularly useful, or maybe not the most cost-effective way to do something, if it's not making things worse, then is it a problem? Right? If it's providing that grower with personal satisfaction, they like that system, they like that approach, that rationale, then is it a problem? Probably not. However, there are times when I'm like, particularly with newer growers or growers with maybe less financial cushion, where I get nervous because I'm like, okay, well, if you're spending time and money, and time is money, on some approaches that maybe aren't going to give you the best bang for your buck, then I get a little concerned. And then similar to what you were saying, I have kind of had conversations before where I've tried to relate it to cancer in people. We know that cancer is multifaceted. There's some cancers that certain things that you have done or certain environments that you've grown up in may increase your risk of that cancer. But also some of them are spontaneous mutations. Some of them are genetics. Then, of course, there's interactions between all of that. And you wouldn't go to a cancer patient and say, if you had only balanced your diet better, you could have avoided this. That's maybe a drastic comparison, but I do feel like there are some folks who start growing and then they get this feedback and they're like, I am having flea beetles in my brassicas. What am I doing wrong? Why do I have flea beetles hitting my brassicas? Whereas anybody that's been growing for a long time will be like, oh, it's because you have brassicas. And flea beetles love brassicas. And that's not to discount like the truths that are also part of it, right? We often talk generally about how a weak plant will suffer more from pest and disease. It'll be more prone to it. What I always kind of wonder, I don't know that anybody could ever test this really, but say you could take away all of your weak plants. Does that mean the insect pest or the disease pest won't be present at all? Or will it then just move on to whatever is the next weakest one that it finds, right? If you have a pest species that is highly targeted to a crop family, and you only have resistant members of that crop family, the least attractive to that pest, they still need to eat something. And so I assume they're still going to go for it.
100%.
That being said, I've never seen cucumber beetles on my loofah plants, right? They just don't seem as interested. They go for the flowers, but I don't see them feeding on them. But then again, I also always have cucumbers and squash, which they'd rather be getting into. So did I balance the nutrition on my loofah plants? Or is it just that they would much rather feed on the cucumbers and the squash?
I really like your cancer analogy, and I think we will probably return to it as we work through what I have planned for you, because it's really nice to come back to, like, what does it all mean? It's not myth busting, right? It's like, these are real phenomena that are happening. It's just not a robust enough phenomena for you to consider seriously. Like, it's real, but it's not helpful.
Because I've been trying to think through this for years. I've encountered more approaches and ideas than I expected, I guess, or whenever there's a disconnect for me personally. This doesn't line up with my understanding. Am I wrong? Am I partially right, partially wrong? Does it matter? Questions along those lines. I think there's a lot of psychology, which those of us who went to school for plant and soil science, we didn't get into that. It always feels better to do something than to not do something. So if you feel like you're mixing up a really great spray and you're going to go apply it, it feels like you just did something. How can that not be a benefit? Whereas sometimes it may be just as good to have sat and waited for the weather to get better. And then that may have allowed soil health to do what it needed to do and correct the problem. And maybe that's probably not that black and white, but
yeah, it's all important to lay the groundwork for how we're talking about this, because it's not just like we can pick apart the details of these claims. Like, there are some added emotions. Like, we want to be open and skeptical, like copious amounts of openness and skepticism. That is something I learned in science school, right? And in this case, we are being very open to ideas and equally skeptical. So here I am. I'm going to make the argument that even if you could manipulate your fertilization strategy in the way that this particular resource suggests, you would still have pest problems. I want to come back to whether or not you actually could manipulate your fertilization strategy in the way they suggest. So we've kind of established this concept is part of the cultural zeitgeist. But for today's conversation, we are picking one resource and focusing on that. We're not going to name the resource, but it was a good starting place. One little thing that I noticed about this one particular resource is they start with a reference that's so old that I was like, that's an interesting choice. Like, what gives there? Because they jump from a very, very old resource to a modern resource. And I'm thinking, oh, like, I can kind of get the allure of going back to like, how did people used to do it? Is that how you read that too? Or do you think there's a reason they started with a really old resource?
I think it's very common to do that, especially in the world of either quote unquote official, like certified organic, or just folks who are similar philosophy of growing, of lower inputs, maybe trying not to spray pesticide if they don't have to. A lot of folks will reference USDA publications from prior to the advent of most common conventional pesticides. So you'll get some really old documents that people really like to go back to, because it was a time when we didn't have cheap synthetic fertility, we didn't have a lot of synthetic pesticides. And so, yeah, how were people figuring out what were the best things to do? I think a lot of folks also see the late 19th century as like the heyday of small yet commercially viable farms. And whether that's true or not, but like here in New England, you can kind of picture that farm where it's not just a homestead, they're producing for themselves, but they're also producing surplus that they're bringing to market. And there's no huge highly efficient operations that they're competing with. So their market is more favorable to them.
Okay. So then they jump to a more modern review paper, which I read and it was really cool. I want to share a couple diagrams with you. Did you know that my expertise in entomology actually is hemipterans? Fun fact.
I did not, but
yeah,
I'm also not shocked.
So here's a diagram where they are showing how aphids feed. So you see this little aphid, she sticks her proboscis down into the cell, and this is a way blown up diagram where you see that she can actually feed her proboscis between the plant cells until she finds phloem sap. This is buckwild. Something that I have been trained on, but never got a chance to do because I never got a chance to set up my own lab at UNH. They never gave me my own space. Are you familiar with EPG readings?
Give me a breakout of the acronym.
I don't know. It's like electropenograph something. So essentially, it's really quite easy to see how much a beetle has consumed of a leaf, right? You can just measure how much leaf tissue is consumed. But there's all sorts of weird feeding behaviors that we want to know about when it comes to these hemipteran feeders, because you can't see them. It's hard to measure them. Also, they're associated with transmission of a lot of pathogens. So there's certain feeding behaviors that are associated with transmission of diseases. For example, there's another group of leafhoppers that transmit bacterium, and there's like certain feeding behaviors that they do that you can actually map in these waveforms. If they don't do that feeding behavior, basically like a clearing of the throat, like into the plant, then they haven't transmitted that pathogen. And so each of these little waveforms is recorded. They basically, and I've done this is so adorable. You glue a little wire to the back of a little tiny insect and you put it on the plant and you run like a really little tiny electrical current that completes the circuit when the insect touches the plant, right? So you're running this electrical circuit, and any kind of little disturbance in that electrical circuit ends up with some of these waveforms. She's putting her little proboscis and feeding it through the plant tissue. And like once she finally hits phloem, when she starts feeding, there's all these waveform patterns that over years and years of work, they have something called an EPG library. So they can say, when you see this waveform, it means she's sucking. And when you see this waveform, it means she's doing X, Y, or Z. All that to say, a lot of this paper is talking about things in terms of understanding how hemipterans make host plant choices based on their feeding. And this is really different from other insect groups because most other insect groups make some of those choices well before they get to their host plant, or there's more choices going on. So if you think about a moth or a beetle, they can use odors to either smell their host plant, smell other insects, their friends, and the smells that are coming from their friends to help them get to their host plant. That's not the case with most hemipterans. They don't have as robust of a smell ability, right? So their approach to host finding is kind of like, do you know those like little, it's like a little baby toy and it has a bubble and there's like little balls inside and they bounce all around? That is kind of how hemipterans find their host plants, is they bounce all around, they land on something green, and they like actively growing tissue. Like you said, like that's their favorite. So that's why a yellow sticky card is attractive to them, is the wavelength of yellow looks more like actively growing tissue than green. So they'll land on it, they'll take a test probe, and that test probe will indicate whether or not they feed, how long they feed, and in some groups, especially some of these leaf hoppers, how far away they jump. If it's a very bad host source, they'll jump further, which is so adorable.
Okay, this makes sense for the mobile hemipterans. Are aphids the exception that make the rule? Because then a lot of them like show up and they are not particularly mobile.
Yes. And so there is another paper that's presented in this particular resource, and I really want to go through that. Your point's a really good one, because how researchers think about pest resistance, right? It's in one of a few different ways. Some of it is something that happens just inside plant cells. So you're really only thinking about pathogens. So like one defense is like a firewall defense, like they just shut it down. So if a cell, like one cell detects a pathogen, they will produce reactive oxygens, basically just like hydrogen peroxide. They will kill that cell. That's different from what we're talking about. So for insects, we're talking about anti-zoonosis, which is non-preference. So that's like an insect flying around, landing, deciding. So like that's when we're talking about winged aphids. So there is a moment in the life of this where aphids grew wings and they decided to look for a new place to be. And then you have antibiosis, and that's like the aphids are already on the plant, but they are either not thriving or there's something happening to them that's making their population go down. And then there's tolerance, which they don't talk about tolerance at all, but I do, if we have time, we'll get to tolerance. So this other paper, this was actually really cool. One of the most buck wild things about this claim, and I want to make this really clear, is that, and maybe this just hurts my feelings because like I'm an entomologist, and anytime I hear people refer to pests broadly, like I think your example of the flea beetles was a really good one, because when they're talking about quote unquote pests and they're just talking about aphids, that doesn't count flea beetles. So like the assertion that you don't have to worry about pests because of this thing about something very specific about one specific aphid species, that's a buck wild assertion to me. And I can understand why people might do that. But like when we're talking about aphids, there's like 5,000 species of aphids.
Yeah, I mean, I was even just kind of like glossing over the fact that really the flea beetles on your brassicas are probably cruciferous flea beetles and not any of the other many species like the ones that go wild on my tomatoes.
Even within those cruciferous flea beetles, there's a couple different species we're talking about too. And even if we're talking about like the 500 species of aphids that exist, even if we're just talking about the aphids that are economically important, there's 100 species of aphids we're talking about. So the paper that they cited, it was really cool, and we're going to work through it. But they are specialist aphids feeding on tansy, which I didn't even know what tansy was. I had to look it up. It's a flowering. Did you know what tansy is?
People use it as an herb, I believe.
Okay. Essentially, what they did is they were growing these tansy plants hydroponically, which is really important because it allowed them to control nitrogen inputs. Granted, they only had control over what they put into the plant and not necessarily what they got out. They did measure it, and they did something really cool to measure it. They had 4 different treatments, 2 treatments that were like kind of within the normal range, so 1 and 3 micromolar. Then they had 2 very high ranges, so 6 and 12. So they had normal range and then like double or quadruple the amount of nitrogen amino acid sources. They had 2 different specialist species, and they did find that aphid species settled 2 to 3 times more frequently on plants that were treated with the high range compared to the normal range. This is something that we know, right? This is not that. But to take that and say, oh, if you just stuck with the normal range, you wouldn't have a problem, that's wrong. And even this paper really shows it. But first, I want to tell you how they measured the amount of amino acids that were actually in the plant sap. Because the folks who are interested in plant sap, they're testing plant tissue for the amount of what carbon to nitrogen ratios by sampling the whole leaf. Is that how it works? Do you know?
It depends on what you're doing. I mean, I think also maybe this is a moment to take a step back and sort of talk about nitrogen measures of crude protein, actual protein, and the peptides and the amino acids that make up protein. Because a lot of the time, when we're talking about plant tissue, if you're thinking about like a livestock person who grows hay, one of the critical values you want to get tested is, you test for your fibers and you test for your crude protein. And a lot of folks don't realize that the test doesn't actually count the protein in that hay, right? It just counts the total nitrogen and then applies a multiplier, because there's lots of different forms of protein and it's made up of a bunch of different peptides, which are all made up of individual amino acids. But when you average out most protein, pretty much most of what's alive, you can sort of get really close by applying a multiplier that you get your total nitrogen times 6.25, if I'm remembering correctly. And that gives you your crude protein. And that's why it's called crude protein, because it's not a measure of actual protein.
But it's good enough. It's a good enough stand-in.
Yeah, it's totally useful.
Well, here, let me show you a diagram. The way that they measure the amino acid concentration in the sap is they allow these aphids to start feeding on the plant so that the aphids are tapped in. And they take a cautery tool and they slice off the mouth parts. So they're basically cutting off the aphid and leaving the mouth parts in the plant, which is delightful. I don't know. It's gross. It's amazing.
Using aphid mouth parts to create a straw.
Yes. And so they're successful in doing this, that kind of drips out and they collect it. As long as that plant is actively moving stuff around, it'll drip out. So they're collecting this exudate from those aphid mouth part straws. So they said 1 and 3 is normal, 6 and 12, they're blowing it out of the water. That's the limitation of, we have these 4 treatments, but the first 3 are kind of the same when you look at how the plant actually is moving stuff around. Do you have any insights to that? Am I interpreting that correctly?
It sounds right. I think what I would see this as is that you see when you give it that much, the highest rate of nitrogen, that the plant has more nitrogen than it can create carbon to use it. It gets really tricky because it's what is your denominator? You're measuring something. And then if you have a healthier plant, it's bigger. And so there's more dry matter that it's being measured in relation to. In that instance, what is your total nitrogen? If you're following a molecule of nitrogen all the way through, it's a little different than if you're just looking at this concentration. So this is on the basis of a certain amount of sap. If you have a very healthy growing plant and photosynthesizing at peak efficiency, it can only create so much carbon, and then it will pair that carbon with nitrogen to make proteins and make new growth.
Okay,
but
cool.
If you have a ton of nitrogen, you're just going to start building it up, and then the plant's going to throw it into a few different amino acid forms to make it not toxic.
Awesome. So that means that the authors that did this work, they found the right range of treatments to test what they wanted to test, right?
Yeah.
So really high levels of these excess amino acids in the plant sap. So here, I'm going to show you the next figure. We're talking about 2 different ways of this changing how aphids interpreted these plants. It's do they light on them? Whether or not they were successful reproducing once they got on these plants. All right, can you see this? This is the reproduction rate of aphids per day.
Is that 2 species of aphids?
Yes, so there's 2 specialist species of aphids. Of course, I've never heard of these aphids before, but they are specialist feeders on tansy. And this is their reproduction rate on these plants. So we just demonstrated that 12 has much higher rates of amino acids versus 1. And the reproduction rates are not different.
Step between your second to highest and your lowest rates of fertility. Those are technically different.
Oh, yeah. So that difference between 3 and 6, that's interesting. It doesn't really line up to the amino acids.
It suggests that actually an optimal food source is one that isn't exposed to that really high nitrogen. That's what I'm saying, is that you don't want that much nitrogen in the system, because I wouldn't be surprised if the plants in that highest rate don't have much available sugar for the aphids.
Yeah.
because all that sugar is being used up for new growth because the plant's going like super fast because it has as much nitrogen and it's not limited by nitrogen at
Yeah.
all.
And then finally, one more figure. So here we see these same treatments again, and we have number of aphids after 2 days. So they're seeing that, especially in this one species, you see a lot more aphids on the treatments that have higher amino acids, right? That proves the point, right? This is something that we know. There's a relationship between the success of amino acid limited species on plants that have higher amino acids. But if you look at the treatments that have the normal rate, that's 100 aphids on that plant. Is that a pest density that we're okay with?
Not usually. My question also is to get kind of back to the what is your denominator point. Do you have more aphids on the plants getting more nitrogen fertility because they're bigger plants and there's more plant matter to support more aphids? And it may actually be the same density of aphids per square meter of leaf. This is the problem with us as scientists, is that we're always like ready to consider it another way or think through it. And that's why we're not giving the full-throated rejection of claims that causes controversy and interest on the internet.
Well, actually, I do think I have a full-throated rejection of this. So I have a couple of examples for you that may or may not help with your point there. But taking it a step further to say, could we alter the nutrition of the plant in a way that limits the growth of aphids? And if you go back to old thinking, at one moment in history, we did think that there were essential nutrients for aphids. So there was like a list of 9 amino acids that could not be synthesized by aphids. They had to get them in their diet. So in theory, if you didn't have those amino acids in their food, then they would suffer and they would not thrive. However, we have since learned that aphid have symbiotic bacteria that synthesize amino acids into essential amino acids.
Well, I think we could even pretend that we didn't know that there were bacteria that can help synthesize amino acids, right? Because if you are trying to have plants that don't have those essential amino acids, I'm guessing, I don't know exactly which amino acids they are, I'm guessing most plants also need those amino acids for good growth. And that if you're not supplying enough nitrogen or there's not enough in the environment that it's growing in to create those amino acids, then the plant is also not thriving. It seems like that would be on the face of it the way it would go.
I guess my question for you is pretty easy to answer them.
I do. But are we basically just saying that, yes, the higher nitrogen plants might have a higher success rate of the aphids, and you might see more of them on plants that have that like lush growth, that rapid, weaker growth. But if you kind of like that earlier thought of like, if you got rid of all of your weak plants, would you get rid of the pest, or would the pest just move on to the next plant?
Yeah. Yeah, exactly. Like that's a really good example. I have a couple of examples where this phenomena that we know to be true, right? Like it's not like you need to cite the paper. There's dozens of papers that there's a relationship between overfertilization and the success of insects that are on that crop. So there is a study that was done by folks out at Cornell who are looking at onions and thrips. So this is a completely different group of insects. These are not hemipterans. These are thysanopterans. But here's another insect group that is documented to thrive on overfertilized.
Instead of the fancy mouth part that can thread the needle between cells and hit the phloem and get that sap directly, the thrips have the rasping and they're like shredding cell walls and getting plant juices that way, right?
Yes, thank you. So they did find a positive relationship between thrips population and overfertilization in this study, but no difference in outcomes, right? So there was no difference in yield or quality. And that gets to your point, which is like, yeah, you can measure differences in populations, but like if it's a bigger plant, if it's a healthier plant, yada, yada. Another thing that they brought up when they did this, they were looking at thrips-resistant cultivars. So they took it a step further and they were looking at different nitrogen regimes same. So kind of they took the same approach, like normal range, high range. The cultivars with resistance had lower levels of onion thrips. However, those same varieties who had lower thrips also had to 10 times more bacterial bulb rot than the thrip susceptible cultivars. And depending on what your problem is in the field, like if you have bulb rot, you are absolutely not going to go with a thrips resistant variety because it's more susceptible. So to your point, like it depends on your situation.
Or at least those ones were. I don't want to shut the door yet. Maybe there's a thrips resistant variety that is also resistant to bacterial rots.
Maybe, or if you don't have bacterial rots in your field, then that's a good option for you. I do have an aphid example for you, if you want me to talk about that.
Sure.
So, you know that part of the reason I know about aphids specifically is that we were looking at aphids on winter-grown spinach. So this is during a time where there are no natural enemies to keep these aphid populations in check. So especially here in New England, aphids are almost never a problem. And I say that very boldly because the only times I've ever seen aphids become a problem is if you are excluding their natural enemies, if you are killing your natural enemies with pesticides, or your natural enemies aren't present because they're asleep, they're dormant, right?
Or you're growing in a high tunnel and they can't get in.
They can't get in, right?
Undercover seedlings in greenhouses, they can become issues.
100%. And what will happen a lot is that growers will start their spinach in the fall. They'll cover them up. They'll go through the holiday season and not really think about it because their spinach plants are just hanging out. And then like shock horror, they uncover and they have aphids. So we were looking at biocontrol remedies for this. And here's the embarrassing part about this is that when we first wrote objectives, we stupidly thought that, oh yeah, I'm sure there's a threshold somewhere. Like whenever you read about aphids, you say aphid feeding will impact yield and quality. This is something we say all the time. It's kind of folklore at this point. For the spinach growers, their thresholds were much lower because it's a crop contaminant. So if they're washing that spinach when they're harvesting it, as long as the aphid populations aren't too high, they'll wash them off, no problem. What we were trying to say is like, oh, I think it was an economist was asking us, at what point does the aphid population get so high that it's actually impacting yield? And we're like, oh, we're sure we can find that. There weren't any references about that. And so we tried to answer that and we couldn't because the plant responds in such a way that it compensates. I can share with you a paper if you're interested in this, but essentially Sam Weldon, she says in this paper, for spinach, there was little evidence that aphid accumulation caused a yield by weight reduction, but rather there was a positive effect of aphid accumulation on spinach biomass. So the more aphid feeding, the more spinach they had, which, what?
I mean, I think you'll probably get people who just have a hard time stomaching that, thinking that that's true. But to your point, you said yield, and the issue is quality. We don't want distorted leaves. We don't want aphid bodies that are a real pain in the butt to wash off. And you don't want aphid bodies going home with the customer and having them deal with it. And so it doesn't really matter if the yield isn't affected, if it's still not working for your system because you've got a bunch of aphids.
Not to tell you how the sausage is made too much, but we, for a very long time in the lab, were trying to answer this, right? So we had single potted spinach plants in a growth chamber that was set to winter conditions, and we were trying to put different concentrations of aphids on it to get to a point where we could say, at what point do you see that weird growth? At what point do you... We couldn't do it because there was too many aphids. We couldn't count the aphids. So after a certain point, we gave up and we said, no, we're going to take your approach and say, what's the point? And we were saying, let's develop a threshold that is like, if you start seeing aphids in your third wash, if you're triple washing, that's when you need to do something. Right? We're not scouting in the field. We are not trying to measure it in the lab. We are saying, how do you actually visualize these things? And anybody who was doing a triple wash, if they were seeing aphids in their final wash, then they should be doing something in the field.
Is this before or after we talked about my grower that was talking to me, who was wondering parasitoids because aphid mummies were more difficult to wash off the greens?
That's bothered me for a really long time. Anyway, do you find any of this satisfying? Did this answer any of the question marks that you had in your head about this?
Yeah, we should bottom line it, right? I think there's several questions. One, is it true that aphids are feeding in response to hit an amino acid threshold? There's theories that lots of different animals are feeding up until they meet some sort of protein requirement, and that if they have less protein-rich food, they need to eat more of it. And if they have very protein-rich food, they can eat less of it to meet the same protein need. And so I think, yes, you're clarifying that, yes, aphids do need to get a certain amount of amino acids. And even if they're not the essential ones that they need, the bacteria that are already in their guts will help to transform them into the specific amino acids they need. Then the next question is, can we confirm that higher amounts of nitrogen, over-fertilization rates of nitrogen, can lead to higher aphid problems? Yes. And then the next question is, should we try to reduce the nitrogen status of our crops in order to reduce the aphid issues? And the answer is probably not, because it's unlikely that you can do that without impairing the crop's growth before you have any real impact on the aphids. Like, who cares if you have aphids if your crop sucks?
And who cares if you have aphids if your crop is great and you can just wash them off?
Right. And if you are seeing big amounts of aphids outdoors, did you just take a row cover off? Because usually we have enough wild predators and parasitoids to keep their populations in check. Sometimes you'll see pockets here and there. For a small scale grower, for a home grower, I am like, get that hose out, knock them way back, and that'll just buy you some time for your predators to come back.
Well, I'm going to leave it there. Thank you so much to Caleb for this conversation. And thank you to you for listening. Find photos of what we discussed on our Instagram at Serious Grower. You'll also see the figures that we discussed at length. Serious Grower Podcast is a production of New Hampshire Community Supported Research. If you are interested in becoming a co-host or would like to learn more about our mission to democratize agricultural research, visit our website at nhcsr.org or come over to our YouTube channel at NHCSR, where we're sharing insights from the agricultural research community and vlogging about our research efforts on the farm. You can support this work by liking and subscribing, and even better, consider joining our Patreon community.